Our session today. Hello everyone. Hope you're all doing well. My name is Zahid Asghar. I will be the instructor for this workshop. Before we begin today, we just like to give a brief moment of silence, a special acknowledgement for Professor Rafa Siddiqui, who was the president of ACIT. He served as the president of ACIT. He was an excellent scientist, and it pains us to say that he passed away a few weeks ago. He was also my teacher as well. So we just like to give a brief moment to recognize Professor Siddiqui before we begin today's session and all that he's contributed to ACIT and to the scientific community at large as well. Our what's it called session today is a summer crash course on meta-analysis. We will specifically be looking at in silico techniques as well, using the program that's known as IPA from Kaizen. Our later sessions will be covering exactly how to use Kaizen and their software called IPA. It stands for Ingenuity Pathway Analysis. But today's session is more of an introductory lesson. So we'll be going over what meta-analysis is, the different types of studies, and just to give you a foundation onto like what the different types of studies are, so that moving forward you have an understanding of the significance of using IPA, the benefit that it gives when it comes to conducting meta-analysis and in silico studies in general. So our sessions will be running on August 3rd to the 31st, every Saturday at 11 a.m. Eastern Standard Time. And if you have any questions as well, you can just write it in the chat box, and we'll leave around 5 to 10 minutes at the end of the session in order to address any questions. And there will be a quiz at the end of the session as well, a 5-minute quiz. So just so you're aware of that, there will be a quiz at the end as well. Okay. All right, so let's just talk about the research hierarchy to begin with. So you may have recognized, you may remember seeing this image on the brochure for this session, for this crash course. So this I just wanted to add in order just for your understanding sake, right? To understand like what the different types of research are and the significance that meta-analysis plays in these different types of research. So you can sort of think of this as a scale of the importance or significance of different studies. And the higher it goes, it has greater significance and quality in terms of research. And the lower it goes, has less significance, and there's increased chances of bias as well. So we can see at the bottom of this list, we have editorials and expert opinions, right? These are just more so like theoretical, they're not actually based on any sort of empirical evidence. They're, not any sort of experiment was done, no study was done in this. It's more so just theoretical. Then we have in silico analysis, which is what we'll be looking further into this workshop as well, with the use of IPA and technology usage. The reason why this is lower than the other types of research that are further above, like case reports, cross-sectional, case control, cohort randomized controls, that it utilizes technology to basically predict how something will happen based off our knowledge of biological processes and such, but it's not actually conducted on any humans or animal models or such, right? Or laboratory studies. Is this sort of like a simulation of how we expect things to happen? So because of that, we've put it below case reports, cross-sectional, case control studies, cohort studies. So above in silico, again, like we have the case reports, cross-sectional, case control, cohort. These are all types of observational studies, which we will be explaining later on in the study and in this session. And above that, we have randomized control trials, which is a type of experimental studies. We will be explaining the different types of experimental studies as well. And on the top, we have meta-analysis and systematic review, which are both, we can consider both of them to be the most significant of the research types that there are. And it's important to note that these have impact on healthcare systems, meaning that case reports, cohort studies, right? They do play a significance in determining, like, for example, risk factors for different diseases, right? That's something that you can determine using a cohort study or a case control study. So they do have an impact when it comes to the healthcare setting. So we'll just begin, so you have an understanding of what the different types of research are. And, we will be getting later down into explaining like why systematic review and meta-analysis are considered the best of these different research types. All right. So broadly, we can classify research into observational studies or experimental studies. So the observational studies, like I mentioned in this previous slide, we have the case reports, cross-sectional uh, case control, and cohort studies, right? These are all observational studies. And basically the difference that you should know about observational versus experimental is that observational, you just let things naturally flow as it is. You have no sort of intervention that you make. As opposed to experimental studies, there is some sort of intervention that's made. For example, that could either be treatment, that could be some sort of surgical intervention, the list goes on, right? So observational, just let things go naturally as they are, as a very rudimentary definition. Experimental, you make some sort of intervention, be that treatment, be that some sort of any sort of intervention, right? So we'll just get into the different types of observational studies that we discussed before. Actually, before that, like we'll talk about editorials, expert opinions, right? So these would not be classified under either observational or experimental studies because they're not actually founded on any sort of empirical evidence. They're just sort of the opinions, excerpts from professionals like doctors, researchers, right? These are more so opinion based, right? They're the opinions of professionals that are, they're well versed in their fields, but there was no actual study that was done to back these findings, right? So these, can include things like court testimonials, right? You have a researcher or a physician testifying in a court testimonial, health emergency protocols, right? So things such as these, like you can even add into maybe the COVID protocols, right? Not all of it was necessarily backed by any sort of study, right? It was just the expert opinions of professionals in the field, right? So this is to note that editorials, expert opinions, they will always be inferior to any sort of study that involves either observational studies or experimental studies. So these are at the bottom of our research hierarchy. So the different types of observational studies that we'll be discussing, these are case reports, case control studies, cross-sectional studies, and cohort studies. So I just have this timeline right here, just so you have like a basic sense of like how these different types of observational studies sort of work, okay? So case control studies, you can basically think of these as you're looking at past cases that have already happened, right? And you're trying to see, okay, what sort of led up to them developing the disease or the condition that you're looking at, whatever you're looking at. Cross-sectional are just sort of like a snapshot of the current time. That how does it look right now? Say, for example, you're looking at diabetes. How many patients currently, or how many participants currently have diabetes, right? It's just sort of looking at the current state of things, the current prevalence of things. Whereas cohort studies is that, okay, you're looking at, okay, like does, like, you're looking basically at risk factors, right? So for example, like if we're taking the example of diabetes here, right? You're looking, okay, does a participant smoke? Do they have what's it called, a sedentary lifestyle? Things like that, right? And you're seeing over a period of time, be that 5, 10 years down the road, do they eventually develop diabetes? So it's a difference in like what you're initially looking at. So case controls, you have the participants initially, and you're seeing, okay, what risk factors might have caused the condition. Cross sectional, you're just sort of looking at how it looks right now. And cohort, you're looking at, okay, because of certain risk factors such as sedentary lifestyle or what's it called, sedentary lifestyle or smoking, will they eventually develop the condition that you're looking at? So it's just, that's a basic way they can think of it as. So, and the other type of observational study that we discussed was a case report. So case reports are, so I haven't listed in this timeline, right? But that's because they are just essentially just an observational study that only includes one participant. So they're not, unlike, case control studies, cross-sectional studies, cohort studies, which involve multiple patients. Case report studies are only with one participant. And for that reason, in our hierarchy that we have right here, they are below cross-sectional, case control, cohort studies, because there's not as much as many participants part of it. So that's why they're considered lower than those different studies. And basically, they're used for more for like rare or interesting cases. So, say for example, you have a rare genetic condition, right? Or you have like an interesting finding in a specific disease, right? You'd want to in situations like that, you make a case report on such a case. Now we have the case control studies that we talked about. So these are essentially where you gather participants who already have the case that you're analyzing. And basically, you're comparing those who are cases who have whatever condition you're looking at to controls, which are individuals that do not have the case. And basically, what you're doing is that you're seeing, like I mentioned before, what caused the disease that slash condition in question, and you're looking at potential risks. And these risks in case control studies are called odds ratio. So this is a statistical test that's done to look at what might have led up to the disease or condition that you're looking at. So that's just a term that like you don't necessarily need to know for this session, but just wanted to, for your knowledge sake, right? That odds ratio is the statistical test that you use to look at what might have caused the disease in specifically case control studies. And the other thing is that these are used for more like rare conditions. So, for a rare condition, it's often very difficult to find patients that might have it. So, if the focus of your study is to gather participants that already have it and to compare it to patients that don't or participants that don't have it, it basically makes it a lot easier to conduct a study. If you compare that to cohort studies, right, where you're just selecting for risk factors, right, reasons for why someone might develop it, right, and then you follow them for 5 or 10 years. If it's a rare condition, it's very unlikely for you to actually find anyone, or at least as many participants or any as many cases as you need for your study. So it's beneficial in that sense because you already gather, people with the rare condition, and you compare it to people that don't have that rare condition. So it's useful in that scenario. And the basic, thing, the flow that you can think of this as is that you have, people with diabetes, right? Taking the example of diabetes versus control, people that don't have diabetes, and you look at, okay, what risk factors led to the disease incidence, right? And you might determine that, okay, participants that smoked, had a poor diet, lack of exercise, they were the ones that became cases, essentially. So that's the take-home message you can take from it. All right. And cross-sectional studies, these are just basically determining the current presence of the disease or condition in question, that whatever you're analyzing. And these determine something that's known as prevalence. Prevalence being how many people in a population currently have a condition or a disease. And these consist of your classic surveys or questionnaires. So, basically you have a survey, you ask participants of their disease slash condition status. In this case, we can think of diabetes in this example, right? And then thereupon, you can determine the prevalence that, say, for example, 5% of these survey participants had diabetes, 95% did not have diabetes. So it's very useful in the scenario of determining prevalence in a population. Next, you have the cohort studies that we talked about. And like I mentioned before, these are basically where you gather participants based off their exposure status. So, like, whether they have predetermined risk factors. So you gather participants by that method, and the participants are then followed upon to see who actually develops into cases. So we discussed that for case control studies, the statistical method for determining risk was odds ratio. For cohort studies, it is relative risk. Again, I'm just adding this in there just for your knowledge, just so you have an idea that, okay, for odds ratio right here, we use, for case control, we use odds ratio. And cross-sectional studies, cohort studies, we use relative risk. These are just like different terminologies, that we use. And so like how it basically works, cohort studies, like I mentioned, with taking the example of diabetes, is that, okay, you select for participants that have risk factors such as smoking, poor diet, lack of exercise, and participants that don't have such risk factors, right? And basically, you look at the effect on the disease slash condition outcome over a period of, let's say, 5, 10 years, however long. And essentially, you see, okay, how many of these participants might have developed diabetes, how many did not develop diabetes. That's essentially how it works. And then we have the experimental studies. Now, the two experimental studies I just like to talk about are the randomized control trials and crossover studies. So if you go back, reference back to hierarchy, you can see that randomized control trials are above the observational study, right? And the other one that I'm also mentioning here is just crossover studies. So in general, experimental studies are considered higher significance than observational studies. So we can talk about our first. Before that, we'll just get into the experimental models that we use for these experimental studies. So when it comes to the drug designing aspect of it, right? Like we often utilize computational or in silico methods. So in silico basically means in silicon, right? So silicon meaning computers, right? So it utilizes computational power to basically it aid us in our drug development process, right? Then for drug screening, we utilize cell cultures, which are in vitro methods. So in vitro basically is like test tubes, petri dishes, right? It's sort of the laboratory aspect of drug testing. And in vitro in Latin means within glass. So again, within test tubes, within petri dishes, this is where it's done, and this is more of like the laboratory aspect of it. Then we have our preclinical, which is, we begin with animal studies, which are in vivo studies. In vivo means within the living in Latin. So these are, before we move on to our human studies, our clinical studies, we first have our animal studies that we conduct. And these are, what's it called, done prior to actually our clinical studies. And then finally, we have our clinical studies, which are in humans, which are also a type of in vivo study, but we conduct our animal studies prior to the clinical studies on humans. Right. So we have our randomized control trials that we're going to talk about first, which is the first experimental study that we're going to talk about. So these are, as the name implies, these are just randomized, we randomize the groups that we're analyzing using, for example, a computer software, right? And we're basically comparing treatments slash interventions to participants that don't receive those interventions slash treatments, or also known as placebos, right? So again, there's controls versus treatments. And this is important, right, to randomize because again, like if say you did not randomize, and some participants in one group that are receiving the treatment are, say, all within their 50s to 60s, right, much older, and the placebo group, for example, is within their 20s to 30s on average, right? So the age difference between the two groups can also skew the results. So that's why it's important to randomize the different groups so that there's no inadvertent secondary, like what's it called? uh, there is no secondary reason for why you are seeing the results you are. It is most likely due to the intervention. So it helps in that sense, right? And the important thing that you do with randomized control trials is that they are blinded. So that means that the participants and the researchers, they are both unaware who is a control versus who is a treatment. And this is important because obviously there can be bias amongst researchers as well. If they know that a participant is receiving a treatment, then they may inadvertently expect to see certain results. So blinding is important for that aspect to prevent any observational biases that may arise. Right. So the sort of how the flow goes with this is that, okay, you have running with our theme of diabetes, right? So say you have a new diabetes treatment, right? You can be comparing that to placebo. You could also be comparing it to prior diabetes treatments as well. And you randomly assign them to participants. And basically, you just see what impact it may have had on the participants, determine the level of improvement or no improvement thereof as well, right? So, and comparing that to placebo, you can also be comparing that to prior diabetes treatments as well, right? That's sort of how this goes. And crossover studies, right? Just another thing I wanted to add as well in terms of experimental studies. This is basically where you're comparing two or more treatments or interventions, or you can also be comparing a treatment versus placebo. And essentially, what happens within this is that group A receives treatment A, group B receives treatment B. Then you have a washout period right here. So group A, group B, treatment B, a washout period. Then group A receives treatment B, group B receives treatment A. So they sort of swap. You can think of it like that. And this is also significant. Again, say there's some inadvertent, selection bias. So, having this washout period and swapping the different groups can, like sort of determine if there may have been some sort of selection bias as well. So that's the significance of crossover studies. And yeah, so now we have in silico analysis. So you refer again back to our hierarchy, we did put in silico analysis below case reports, cross-sectional case control studies, cohort are different observational studies, but I'm discussing it here because they do have a significance when it comes to our meta-analysis. So basically, I will be getting into that later down in upcoming slides. So basically, this employs computational techniques and models. So essentially, running models on what's called a computer. It basically simulates biological processes. So our current existing literature, like from databases and such, right, are established understanding of how processes work, right? We use that and we basically simulate that to sort of predict, okay, what's going to happen, and because it's not actually done on what's it called, any sort of animal or tissue sample, for example, right? Or it's not done on humans in a clinical trial. We would like to say that it has less significance than that because it is sort of just a prediction, right? So again, it predicts the effects, targets, toxicities, right? Using our already established understanding of how things work, and it is highly useful for drug design, which I will be getting to later. All right. So we have our systematic reviews. So like we mentioned, systematic reviews and meta-analysis, these two are the most significant of the different studies. Now, why are they the most significant? Is because they employ the results from the different observational studies and the different experimental studies that we just talked about. So they are a culmination of all of those. So you gather all available empirical research by using clear, defined, systematic methods to obtain answers to a specific question. So again, they are a culmination of all of those studies, and you basically culminate all those studies in order to get your results. So it also follows a methodology as well. That's a very important aspect of systematic reviews. So again, you collect, extract, interpret data from peer-reviewed published studies, and it is a protocol-driven comprehensive review and synthesis of data. That is very important. Like you can follow like the PICO protocol, for example, right? That's one of the protocols. Just be perfectly aware that there is a protocol for how you do it. And you analyze, describe, critically appraise, and summarize interpretations into a refined evidence-based conclusion. And what's very important is that the method in which that you obtained your results should be reproducible. So results are reproducible, meaning that if someone went into your methods section of your study, they followed the exact same methods that you had written, they should come with the same conclusion that you came to. So that is very important, how you synthesize your data, inclusion, exclusion criteria, what you accept, what you reject, right? That should all be reproducible by anyone else that conducts the same study, right? And meta-analysis, right? This is essentially the statistical process of analyzing combined results from multiple studies, and you re-examine the effectiveness of drug interventions. So major difference here is that meta-analysis involves statistical processing of the data. So like coming up with things like p-values, z-scores, just be perfectly aware of these terms, right? These are statistical values that you generate from meta-analysis, right? So it involves generating this sort of like statistical data. And much like systematic reviews, these also involve collecting and analyzing data in a systematic method, right? So a lot like systematic reviews, you also collect data in a systematic method. So, again, if your inclusion exclusion criteria, right, is often a part of meta-analysis as well. So an example of a meta-analysis I can just think of is, okay, so imagine subjects from 10 studies, a total of 1,020 subjects were pooled and statistically analyzed to determine the effect of the relationship between diabetes and neuropathy. This meta-analysis showed a 50% reduction in neuropathic diagnosis among diabetic patients with anti-diabetic drugs. So again, what's another important point that I'd like to just highlight with meta-analysis is that the subjects from those different studies are all pooled together. Like you don't look at them individually; they are pooled together into one giant analysis. So this again avoids the need for conducting, for example, like a large clinical trial, right? You're just pooling the results from all those different studies and you're just treating it as one giant study. Okay. And another important point is that oftentimes with meta-analysis, the results from the individual studies may not reflect the results from a meta-analysis. So in a individual study, it might show that the anti-diabetic drug had no effect in reducing neuropathic diagnosis among diabetic patients, right? In an individual um, study. But in the overall meta-analysis, it does show an improvement. So again, that's one thing that you should note, that the results from the meta-analysis may not reflect the results of the individual studies, which is often why it's also considered so significant. All right. So the basic steps that you follow for conducting a meta-analysis. So you select the hypothesis for your meta-analysis. You identify relevant studies on the topic. Then there's the extraction of data. So the sample sizes, the methods, the measures of data variability. And then you synthesize your data for statistical modeling. And then you analyze the overall effect using the statistical modeling, sorry. All right. And now the advantages of and disadvantages of meta-analysis. So advantages are that it's faster, safer, and more precise exploration of new treatments. So, and the other thing, like I mentioned before, it simulates biological and drug interactions, and it reduces the reliance on traditional clinical trials because, again, you're pooling the results of those different trials together. So it reduces the reliance on performing a large clinical trial. It also has utility in predicting carcinogenicity. So it has utility in determining, cancer-causing, substances, right? So carcinogens, it has utility in things such as that without the need for animal testing. And it also contributes to the prevention, diagnosis, prognosis, treatment planning, and execution or management of disease. And the disadvantages to meta-analysis is, although it is better than traditional clinical trials, it is still a time-consuming process, and it does require the knowledge of statistical techniques and whatever abilities that might be required with that, right? And oftentimes it requires the use of sophisticated tools. So oftentimes these may be there may be a cost well because some of these are uh, can be pricey. But so that is a consideration as well that some may make when conducting an analysis. All right. So both of these studies are rather similar, right? But there are some key differences between these two, and both of them are considered, typically the highest quality of research. So, what are some differences? We'll just go over this briefly. So systematic reviews are just basically a systematic method to obtaining answers to a specific question. One thing to note is that oftentimes you generate, a hypothesis as well. You may have begun with your hypothesis, but you often generate new questions with systematic reviews. So that's one important thing to note. And systematic reviews can become meta-analysis, right? And we'll get into how they can become meta-analysis by discussing what the differences between systematic review and meta-analysis are. So meta-analysis, right? These are basically the statistical processing of and analyzing of combined results, like I mentioned, right? And they are the pooling of similar studies, okay? So, that's how a systematic review can become a meta-analysis if there are similar enough studies they can actually pull them together, synthesize them in order to become like in order to create statistical data. A systematic review can become a meta-analysis in that regard. And the other thing is that it increases power of the study as well. So, like we mentioned in this example right here, is that like the subjects from the 10 studies were pooled together. So you have a greater number of participants that you're analyzing your study. And that's sort of, in simple terms, you can think of that as power. So the more participants that there are, the more validity there is in the results. It reduces the likelihood of outliers skewing the results more than they should. So that's the benefit of meta-analysis, that increases the power of the study. Right. So we'll go on to the drug designing process. So stage one is the basic research component. So like when you're first designing a drug, you're looking for your potential targets. Let's take the example of diabetes here. So you're looking for compounds that may have an effect on treating diabetes, right? So you in your initial basic research, you may have greater than 20,000 compounds that you discover through literature search and such, right? That have effects in treating diabetes. Stage two of your study, you'll look for a specific drug target that you're looking for. So, for example, with diabetes, maybe you're looking at something sort of like a diabetes drug known as secretagogues that increase the secretion of insulin from the pancreas, right? Be that through chemical channels, right? So you're looking specifically at drugs that function as secretagogues that increase, like ionic potential within the pancreatic beta cells that lead to the release of insulin, right? So drugs that have that sort of, or compounds that have that sort of effect, that's what you do in stage two, that you look for that specific target, like think compounds that have that sort of specific effect, right? Then in your preclinical research, right, this is your sort of in vitro studies, right? The research that's done within test tubes on tissue samples, right? In your in vitro studies, your preclinical research, you may come down to 250 compounds. So it becomes more and more narrow, right? Then by the time you get to your stage four, which is your clinical research, which is done on human models, done on humans, right? Clinical trials, you may come down to five compounds that you've narrowed down to. And then your stage five, like before drug approval by the FDA, right? You look at more like, I guess, unseen, or like, like you would have otherwise missed, what's it called? Side effects, right? So you determine, long-term effects and such, right? And then you might eventually just end up approving just one of these compounds that you initially started with 20,000 compounds. By the end of it, you only have one that is actually approved for use. So the significance that in silico services play in the sort of drug design process is that it dramatically reduces the time that it takes within stage one and stage two. So, like we said, stage one often involves, collecting over 20,000 compounds often, right? So this process can often take 30 months, right? But with the use of in silico services, that process can be reduced down to days. So it dramatically reduces the time it takes in stage one and stage two as well, which can often take 48 months. With the use of in silico services, the use of computational power, right? Like having the computer basically look at the targets that you're trying to analyze for, right? It can far significantly, reduce that time it takes. Again, because computers, they can sort through data quite a bit faster than we can, right? This process would take months, years for us, right? But for what's it called, in silico services, right? That can dramatically reduce the time it takes in stage one and two, and stage three, four, five. These still are typically done without the use of what's it called, in silico services. These are still the researcher that will conduct these stages, but because stage one and two are dramatically reduced in the time it takes, the overall drug approval process becomes far more expedited with in silico services, right? So the company that, like, is produces the software that we'll be looking into today is Kaizen. So ingenuity pathway analysis, the subsequent session that we'll be getting into will be on how to actually use IPA, right, and utilize it. And the company that makes it is Kaizen. So just like a brief introductory onto what Kaizen is, it's a German multinational company established in 1984, and they have offices in Asia, Europe, and the Americas. So what they do is obviously bioinformatics, like the software that we're going to be using, which is IPA, is part of their bioinformatics section. They also produce molecular kits, pharmaceutical testing, and hardware such as PCR, DNA purification, etc. The list goes on. And the software that they have, their bioinformatics, is not IPA, which is, by and large, is also a database. So it is a collection of different studies, the collection of that empirical data that's been collected, and it's basically the accumulated findings from those studies. So, for example, like if you take, so for example, say a certain compound is inhibited by another compound, right? So that finding is present within IPA, and uh, you can actually simulate that finding from IPA. So, for example, you can create connections between molecules, functions, and etc. Right? And you can use that established, findings of those biological processes in your analysis, essentially. So some of the functions that IPA employs are the grow feature, the path explorer, molecule activity predictor, keep, core analysis, and heat maps. In our upcoming sessions, we will be going over the grow, path explorer, molecule activity predictor, keep, as well. We won't be going over core analysis, but just be perfectly aware that this is another function of IPA. And this isn't obviously an exhaustive list. There are other functions of IPA, but just so you're aware, these are some of the functions that IPA employs. And these are just some examples of analysis that you can do with IPA. This is actually from an analysis that I did. So to the left is employing the Path Explorer feature, which I mentioned over here. And using the Path Explorer feature, I connected ethanol and morphine to pentraxin 3. So, and then I use the molecule activity predictor, which essentially predicts predicted activation or inhibition. So the blue right here is predicted inhibition or decreased quantities. The what's it called? The red is predicted activation or increased quantities, right? And like that is the molecule activity predictor right here. I use that to predict what effect ethanol and morphine might have on modulating pentraxin 3. So yeah, and you can also perform quite complex what's it called interactions as well. So this is another example of what's it called, a study that I did, an analysis that I made. So over here we have ITGAM, which I connected to RALA. So there's, as you can see, there's multiple different compounds that are between these. And basically, you can basically connect multiple different nodes being like these intermediary molecules right here that eventually end up connecting to RALA. So you're able to create quite extensive, what's it called, connections here, which I just wanted to show you. All right. So yeah, we will be utilizing IPA within our third session. The third session will be going more into depth into the utilization of IPA. So during this session, I'd just like you all to apply for IPA. You can follow this link right here, uh, or you can just type in IPA Kaizen on your browser. There's also a QR code that is linked here. You can use your phones, and that will take you to the IPA sign up page. So just to show you, you can all maybe, even after the session, you can maybe copy this QR code to get the website. So basically, once you get onto the website, it'll look something like this. You'll see request a trial here. So you click on request a trial, and it'll take you to this page. You fill in the required fields, first name, last name, company, which can either be the institution you're part of or your school, right? Position, be it student or faculty member, right? Your institutional email address is also required, and you enter your phone number, country, and your interest for using IPA. You can put other, or you could put mechanism of action. You can put, it shouldn't matter too much, right? But you can put other or any other category here for your primary interest for using IPA, and then you just submit. And just one thing that I want to note is that do not activate the trial right now. You should be receiving a trial link within a couple of days. Uh, do in fact apply for the trial right now. Like submit your what's it called, your trial request, but don't activate it yet. That's because the trial only lasts for 2 weeks, and it will be needed for the third and fourth sessions of this workshop, and the activation code lasts for 15 days. So you will have enough time by that point to still activate the trial, right? So I want you to not activate it yet. Request a trial, and at the end of the second session, a few days after the second session, which we will be having on August 10th, I believe, a few days after August 10th, activate it then, and you should have it available for use in the third and fourth sessions as well. All right. Now we'll just have a Q&A session right now. If anyone has any questions they'd like to ask about this presentation or any other questions that they may have, clarifying anything, yeah, you can ask any questions at this moment.
I ask the question.
Yeah, of course.
Sir, please explain timeline in briefly.
Yeah, of course, of course. So you're talking about the timeline for what's it called? The observational studies? This timeline right here. Yeah, so okay, so I'll just briefly explain this again. So basically the timeline, like it's just for like an understanding purpose, right? That okay,
Yeah.
so how these, like, what these different observational studies sort of look at. So the case control studies, right? These are sort of like cases that have already happened, right? Like, say, with the example of diabetes, right? Like, the participants that you've gathered already have diabetes, right? Like, you're not looking at people that just like have not developed diabetes, right? They already have diabetes, and you're comparing it to people that don't have it, right? And you're basically seeing, okay, like what led, like them to eventually develop diabetes, with continuing with the example of diabetes here. So like was it hypertension, was it poor diet, right? You're looking at things like that, right? And seeing like, okay, like what eventually caused them to get it, right? So you start with the cases, right? And you determine the risk factors. That's essentially what you can think of it as. You start with the cases, and then you determine what the risk factors were for them to develop it. And cross-sectional, these are, like, again, like I said, present, right? These are determining the prevalence. Prevalence means how many people in your population currently have something. That's the simplified definition of what prevalence is. So again, like I said, these are your survey sort of studies, right? So you ask participants who has, for example, diabetes, right? And like you just sort of have a snapshot of how the current disease status might look like. That's what cross-sectional studies are. And cohort studies, these are where you first start off with the risk factors. So like, again, with the example of diabetes, look at, okay, who has a, like, like a sedentary lifestyle, for example, right? Or who has hypertension. And looking at, those sort of risk factors, you see, okay, after 5 years, after 10 years, how many of these participants ended up getting diabetes? That's what cohort studies are. So that's just like, the timeline that I wanted to create. So just have like a rudimentary understanding of like what these are. Cohort, you determine risk factors, follow them over time, see who develops the case. Cross-sectional, you want to see how many people currently in the population are cases. And case control studies, you already have gathered cases together, and you want to see, okay, who what's it called? Sorry, what reason led them to become cases? So yeah.
Yeah, okay, thanks. Clear.
Sir, I have a question.
Mhm.
Sir,
Yes.
can you please explain IPA briefly? That it is
Yeah.
not clear to me.
Yeah. So IPA is it stands for Ingenuity Pathway Analysis. So it is one of the bioinformatics tools of IPA of sorry Kaizen. So basically, what they've done with IPA is that they've essentially like accumulated multiple different results from different studies, and like the different biological processes that were established from those studies. So for example, like let's say that they found that ethanol uh, increases an inflammatory mediator like IL-1, for example. Just an example. Ethanol increases IL-1, and this was determined through different literature, right? So what IPA does is that it adds that information onto their service, and you're able to reproduce that on their service. So you're able to like basically have like um, like you're able to have ethanol, and you're able to create a connection between ethanol and that inflammatory mediator like IL-1, right? And you're able to see that, okay, this is how ethanol connects to IL-1. It may not be directly connecting to IL-1, but say, for example, like ethanol activates some transcription factor, and that transcription factor then leads to an increase in what's it called, IL-1. So that whole information will be available from IPA. And you're able to like create like connections like that as like sort of like a visual representation of how these pathways work. So it is pretty useful when it comes to like visually seeing how these connections work. It has utility there.
Okay, thank you, sir.
I hope that was clear. The explanation was sufficient. Yeah. Any other questions at the moment? Let me get back to the chat.
Sir, you said that IPA helps make sense of large amount of biological data, like we get to understand those connection. So IPA, does IPF any other use other than that?
Yes, it does. So like I mentioned, the what's it called? The core analysis feature of IPA. So like I mentioned before, right? There's like statistical values like p values, like z-scores, right? You can basically determine these statistical values using IPA as well. So you can input your own data. Say you conducted research of your own, you can input that into IPA, and it can determine those statistical values like p-values, for example, right? And it gives you something called heat maps. And heat maps are basically like, say you submitted your own research into IPA, it'll basically tell you like the p-values of all the different compounds that you had in your research, right? So it'll process that for you, right? So obviously you can visualize different pathways, you can visualize different reactions and such, right, using IPA, but it also has that statistical usage as well, that you can perform your core analysis, and that gives you all that statistical data as well. So that's another usage of IPA as well.
All right, sir.
We have a role in novel studies too. Could you elaborate on that? Like, what do you mean by novel studies? What's it called? You don't have any prior data when you're what's it called, doing your studies, and like you just want to create a study from scratch. Is that what you mean? Yeah, perfect. So yes, you can in fact do that as well. So like like I discussed with the core analysis. So core analysis, you can also submit your own data for a core analysis. but you can also do it without having any prior data. So IPA also has basically different studies in their database as well, and you can input that data to create a core analysis. But you can also do it without any prior data as well. So that is also possible with IPA. It's not necessary that you have your own data from beforehand. You can also create a core analysis without any prior what's it called data as well. So that's also a possibility. Um, yeah, and you're able to generate all the statistical data that I mentioned, such as like p-values, z-scores, heat maps, that such. Okay. All right. So I guess we can get on to the quiz, I guess. So I will share the quiz QR code. Okay. So this is the QR code for the quiz. You guys can open up your phones and scan this QR code. It should take you to the quiz, like, pin, like where you input your game pin for Kahoot. This quiz is on Kahoot, by the way. And you can just input the code that will be generated on the screen. I'll be sharing the code for the quiz soon. So just everyone just scan the QR code, and it will take you to this page right here, right? Where you have Kahoot, where you enter the game pin in. Once everyone is on there, right? After scanning the QR code, let me know, and I'll share the game pin, and we can start the quiz right now. And just so you know as well, the quiz will have prizes. There will be a quiz at the end of all of our sessions. The quiz at the end will have a prize as well. So that's one thing that you should know. If you attend at least 4 sessions, right, you can participate in the quiz at the end. And there will be a quiz at after each of our sessions. So today there will be a quiz. The next session there will also be a quiz up until the final session. And whoever places first in the quiz receives 3 points towards the final quiz. Whoever places second receives 2 points towards the final quiz. Whoever places third receives 1 point towards the final quiz. So you have more points at the final quiz. So there's a little bit of incentive there to do well on the quiz. So let me know once everyone has scanned the QR code. Let me check the chat. Yeah, was everyone able to scan the QR code?
Sir, can you show your code?
Yes, I'll show. All right. Was everyone able to? Okay. So I'll begin the quiz right now. Hopefully everyone was able to get to Kahoot from the QR code. So this is the game pin right here. All right, has everyone entered in the game pin?
Sir,
Anyone
wait,
that
please.
hasn't? Okay, okay. All right, has everyone entered the game pin? If you haven't been able to enter in the game pin, you can just write it in the chat. Otherwise, I should be beginning the quiz within the next few seconds, I guess. So just you can write it in the chat if anyone hasn't been able to submit it yet. All right, everyone's entered in the game pin. Okay, so we'll begin now, the quiz. I'll be starting the quiz now.
No, sir, wait 2 minutes.
Okay, okay.
Recording in progress.
Sir, could you please tell when does it start?
Yeah, I can start. Just making sure that everyone, there's someone that said that give them a little bit of time to join. We should be able to start once I guess everyone is ready.
Like, I'm facing internet connection, so I might be out.
Oh, I see, I see. All right, let me see if everyone is able to enter. Okay.
Yes, sir.
Okay, yeah. So we'll be beginning now. Yeah, I'll be starting the quiz now. Okay. 23 participants. All right, so we have Dr. Sahil who came in first. Then we have Ahad and then Alfiz Khan. So you each will be receiving 3, 2, and 1 points each. That will be added to the last quiz at the end. Are you writing that down right now? And I hope that everyone was able to, take away from this session. You were able to learn something, I hope. If there was any issue in the way I taught, please do let me know. Any improvements or any such, anything else. But yeah, this is the end of the session. I hope you all were able to take away from this session. And our next session will be on Saturday. Next Saturday, which I believe is August 10th at 11 a.m. Eastern Standard Time as well. I believe that might be 8 or 9 p.m. In India. And I thank you all for coming to this session. And yeah.